Thanks everybody for joining us. I'm Dr. Tanya Brown. I'm the scientific director at TESS Research Foundation, and we have a very special guest here today, Dr. Jen Wang, and we are so excited to have you here and to learn a little bit more about your research. So to start off, can you maybe share, how did you get involved with TESS Research Foundation?
So I was working at STEM as a part of the Resolution Consortium, and I think you reached out to our project manager about the Resolution Consortium and what they were doing. And so then we got to meet, I think, at one of the community research meetings that you guys hosted. And then, sorry, and then
We love furry friends popping up.
yeah, he's playing with the laptop. Um, and then and then I got to meet everyone there. I think Tabea, the project manager, gave a talk, an overview of what was going on. And that's how you guys got to know what we were doing with SLC13A5 in our research. And then I think, and then from there on, we kept in touch and was able to collaborate with some of the people in the community.
Yeah, which has been so fruitful in so many different ways. And that's one of the reasons why we wanted to chat and, learn a little bit more about one of your main projects that you were working on. Can you share a little bit about your SLC13A5 project?
Yes, so we did a deep mutational scan of SLC13A5. And basically what that means is that you're able to look at all the possible variants of this protein, and we assess whether or not they are functional in terms of transporting citrate. And so in this way, we have, experimental evidence that each either transport or cannot transport citrate or to a lesser extent. And so, we did this in cells, and we had a assay for it, and then we were able to look at, I think there are about 12,000 variants in total. And these are single mutations. Yes.
Yeah, thanks for that explanation. And maybe just putting the patient spin on it too. I think one thing that kept this work, even though it's so functional and basic science in the sense of using a cell-based assay, it becomes translational, and that you also focused on patient variants too as some of the high priorities for some of the functional testing, right?
So this was done in a high throughput fashion. So you have you pull all the variants together and you test it out that way. And then we nitpicked 38 variants to test individually so that we can validate the high throughput assay. And so the single variants, a lot of them were patient variants that haven't been studied a lot. And we put that as more high priority.
Yeah, and that has such a big impact for the patient community. And just to share for kind of the broader community why this is so important, in part, is because so many people get genetic reports and they have two variants, one that might be pathogenic or likely pathogenic, and then one that's a variant of uncertain significance. So Jen's data will help say, well, actually, this may have functional consequences, that it's not just uncertain significance, but we can show in a lab setting that there's a loss of citrate transport. And this may be an indicator that someone may be affected by SLC13A5 epilepsy or Citrate Transporter Disorder. So your work is having impact. And I think that what's so great, and one of the things that I really appreciate about you, even when we talked in, gosh, it was a couple of years ago in Austria at the Resolute meeting, Avner Schlesinger spoke so highly of your work and that you are so collaborative. And I just want to say thank you for being so collaborative and open.
Yeah. No, it was super fun. I liked working. Yeah, I like working with everyone in the community. Um, and when everyone's doing this on working on a similar research topic, it gets very collaborative and it gets really, yeah. No, I enjoyed it a lot.
Yeah, well, we enjoyed working with you too. And so maybe that sort of answers my next question, but like what inspired you to continue on this project and really make this something that is so valuable to the patient community as well?
Um, yeah, so it was one of the bigger projects that worked in terms of because with a deep mutational scan, you have you have to have the right assays. It has to you make the library, and it all has to fit together. And so this project, with its ups and downs, was relatively lucky to yeah, and we were able to finish it, and now it's been accepted in Science Advances, and so hopefully we'll see the work soon. Yeah, but everything went quite smoothly in terms of each step, and so it passed a lot of hurdles to um, yeah, to be one of the projects that we managed to finish as part of the consortium.
Yeah, which is
Yeah.
fantastic. Um, I think it's so helpful to hear like this was a win for SLC13A5 for this solute carrier. And can you talk about maybe from your perspective, like what do you think is one of the most pressing challenges or you could say opportunities for the SLC13A5 community in terms of scientific advances and just research and development?
Mhm. Yeah, for I guess for SLC13A5, we managed to do this in just the basic cell line, looking at just transport. So it was basically it's on the membrane, whether it transports or not. But I guess you could also take it further to more relevant cell lines that are maybe neuron related. And you can also look at what it's doing to it physiologically. So not just transporting citrate. Yeah, and so those models, I guess, don't exist yet. Um, and then bring it into like high throughput, looking at all the mutations in that terms, is still, yeah, not there yet. Um, but yeah, hopefully with our study, you can nitpick certain variants you want to look at and look at it in those more disease relevant settings.
Yeah.
And so that would be cool to do.
That would
Mhm.
be really cool. I think
Yeah.
kind of what I'm hearing you say is that, here we're looking at an assay that is directly reading citrate. We know SLC13A5 codes for protein and ACT that moves citrate from outside the cell into the cell. And so we are directly measuring that, or I'm saying you are directly measuring that in this assay. And kind of some of the next steps are like, what are some of the downstream consequences later on?
Mhm.
Because we're looking at cells in a dish and maybe we can understand some of the processes that happen after citrate moves into the cell. Is that
Exactly. Yeah. Yeah.
great? Great. Well, thank you so much. I think, you mentioned that your paper is coming out, and can you share maybe where people can find more of the data if they're interested in learning more about this
Mhm.
assay or have specific variants in mind that they want to look up?
Yeah, it will be available with the paper. So if you go down to data availability, when the paper is published, you can get a link to yeah, to the data deposit website. We tried to make it really simple. It's like an Excel spreadsheet with all the variants, and then you have different data sets associated with each variant. And so you can sort through it. One of the things that would be helpful is to get feedback on whether or not this is easy to read or this is easy to understand. Also for future deep mutational scans that are or data sets that are relevant to clinical settings. Yeah, whether this is translatable to clinicians or at interpreting the data.
Yeah.
So that would be nice to get feedback on.
Yeah. And to give feedback, should we email you? Should we add some
Yeah,
contact or okay?
you can email me or people who are on the paper as well.
Okay.
Yeah.
Great. Well, thank you so much for sharing about your work. And again, it's been so wonderful to get to talk with you, to see you at meetings, to have you present at our summer meeting, last summer. And your work is having an impact already. I mean, I think I wanted to share just there was a baby born with a variant of uncertain significance, and we were able to reach out to you and say, what does your data suggest about this variant? And just how impactful that can be to add a piece of information into this unknown territory. So thank you very much. And to the whole team who's been working on this as well. All right, great. Is there anything else you want to share with the SLC13A5 and TESS community?
No, just thank you guys. It's been great to be part of the community and to hear some of the stories and both within the research field and also with the patients and their families. Yeah, it made the research journey quite a lot and more enriching.
Great.
Thank you.
Great. Well, thank you so much.